Alright, so hopefully this works. Okay, so I'm starting off on the slides that I just posted, so I think it's presentation number 4, version 3, if I'm not mistaken, and right now I'm starting off on slide 15. Hopefully these won't be updated too much more over the weekend, but I'll keep you guys posted if they are. Please comment any questions you have underneath this, and then I'll try to comment back and answer any of these questions as soon as possible. So right here we're looking at slide 15, and we're looking at J and K specifically. As we can see over here where my mouse is, we have J and K1, J and K2, and J and K3. So those are going to be our three main isoforms of J and K. So all J and Ks are serine threonine kinases, and they have 11 protein kinase subdomains, as we can see up here. You can also see the N-terminal lobe and the carboxy terminal lobe. So the protein kinase activation loop, as you can see here, is going to be located between the kinase domains 7 and 8 right here. Oh, don't know where my mouse went right here. So as we can see, that this is where the activation loop is, and that it contains a TPY motif, or threonine proline tyrosine motif, and this is conserved throughout all of the isoforms, as we can see here. So it's all going to be in this general area. Okay, and so we see this, and this is going to be where we're going to get phosphorylation for the full kinase activity of J and K, and this is going to be by MAP2Ks. So as Tosh explains in some of her slides earlier, we have these things called MKKs, or MAP2Ks is synonymous with this. And whenever you see something that says like MAP2K or MKK, however many Ks there are, that's going to be a kinase of a kinase. So J and K is an example of a MAPK, whereas an MAP2K would be a kinase of the MAPK, and a MAP3K would be a kinase of J and K or other MAPKs. So hopefully that makes a little bit more sense. So a MAP3K or an MKKK is going to phosphorylate an MKK, which will phosphorylate an MK such as, or a MAPK such as J and K. So again, just to sort of get rid of any discrepancies we have right off the bat. So you're going to get J and K activation by serine threonine, inactivation by serine threonine phosphatases. And specifically, this is all just I'm reading from the notes here, is that J and K1 and J and K2 are found in cells and tissues, whereas J and K3 is only found in brain and cardiac cells. So differential splicing of J and K1, 2, and 3 will result in the different isoforms. And the two splicing sites are between domain, oh my gosh, I can't read Roman numerals, 9 and 10 in the C terminus. So as you can see here, we're going to get differential splicing in between here, as well as at the C terminus here. So you can see how they have different lengths here. So that's just differential splicing. So we tried to go more in depth about, so we're looking specifically at J and K1. As it relates, we see it how in the second presentation, I believe that we saw that serine phosphorylation of IRS1 is going to be by J and K. J and K1 specifically, we looked in to try to see what specific isoforms of J and K1 were involved in specifically like diabetes and hyperglycemia, and we figured out from an outside source that it was J and K1 beta 1, but like we literally can't find any information on it, so stay updated for any new things on that. So I realize I'm going really slowly, so I'm going to hurry it up a little bit. So in this photo here, we can see the J and K1 structure. Right here, this little purpley blob, it's coupled with GIP1, which, as Tash will explain when she posts her video, GIP1 is going to be a scaffolding protein that's going to be responsible for sort of combining all the MAPK kinases together and allowing J and K1 to be phosphorylated and in turn activated. So here in this JNK1 structure, we see an N-terminal domain and a C-terminal domain. I have here that we have 7 in the N-terminal domain. I just explained the amount of beta strands and alpha helices. I think you guys can read, so hopefully that's clear. I don't want to waste too much time on easy stuff like this. In the C-terminal domain, we have the same thing again. We also have these 310 helices that come up again, which I know we had when I presented, and Abby said that it wasn't a form of secondary structure, but it is a form of secondary structure. I added in the notes a link to a wiki page just for you guys to refresh your memory on what those are. So we also have connector loops, and they're going to be between B7 and B8, so right here, as well as between A13 and A14, which, if I can find it, A14 right here and A13 over here. So where they have these green dots just means that they're not really sure what's going on in this part of the structure. JNK1 is pretty flexible because of these two connector loops. And again, this was the only article I could find on JNK1 structure, and it's from 2004. This article was originally talking about how JIP1 can cause the inactivation of JNK1, which is kind of silly because later articles and years later they sort of realized that, hey, like JIP1 is necessary for the activation of JNK1, so it can't inhibit it. So saw some discrepancy there, but it was really the only article that showed the full structure of JNK1, and so this is what we're going to use it for. So JIP1 doesn't inactivate JNK1 unless it's really overexpressed, so the article was wrong in that sense. So hopefully that's clear. So JIP1, along with the MAPK kinases, come together and phosphorylate JNK1 so it can be activated. So I just skipped slides here. They're pretty much the same thing. We talk about these 3 specific domains within the JNK1 structure. Again, very, very vague as to where these actually are. As you can see, we have a couple residue names, but it was super vague, so this is just sort of my interpretation of what's going down here. So we have 3 main sites. We're going to have our activation loop, which is going to contain our threonine, proline, tyrosine motif that we talked about before. It's going to be really, really important for JNK1 activation. The ATP binding site is going to be, well, okay, this is what the ATP binding site was said in the article that lies between the N and the C lobes, so somewhere in here, whether it's on the first connector that we talked about between B7 and B8, or if it's between the second connector we talked about between alpha 13 and alpha 14, I could not tell you. After reading this article a bunch of times, I'm not quite sure. So yeah. So what we have is we have the ATP binding site, and it said in the article that it's bracketed by the A loop and the peptide docking site. Peptide docking site, another word for it that we're going to use is common docking domain, or CD domain. Okay, so yep, so the ATP binding site is in between the two. That's why I ordered them in this way. So the ATP binding site is a pocket formed by alanine, so 3 residues in the N domain, so up here, as well as 3 residues in the C domain down here, as well as in between the linker loop between the 2 lobes. So as we can see, we're not quite sure. Apparently ATP binds essentially in, kind of gets sucked into a pocket that JNK1 forms. So then we also have the common docking domain, which is a cluster of negatively charged amino acids, and it's going to be more so in the it's going to be in the C-terminal region of the kinase domain sequence, which I'm taking to mean the activation loop. So again, the common docking domain is going to be, we're going to get the activation loop, which is going to be somewhere higher up than the ATP binding site in this general region, and then we're going to have the common docking domain down here. So hopefully that makes sense to you guys. It's making sense to me, so hopefully it makes sense to you. So the structural features that determine J&K specificity preference for different D motifs are currently unknown. So any substrate that's going to bind to JNK1, whether it's going to be JIP1 or a MAPK kinase, is going to have something called a D motif, and this D motif is going to bind to the CD domain, and that's how the two substrates are going to bind together. So J&K's specificity preference for different D motifs isn't really well known, but we do know that D motifs on different substrates that bind to J&K contain hydrophobic motifs that are 3 amino acids long. And yes, GIP1, they say that they don't know the different specificities, J&K specificities for different D motifs, but they do know that it has really strong specificity for GIP1 motifs. Okay. So this is just going to show a quick slide showing the interaction between J&K1 and PEP-GIP1. So PEP-GIP1 is just a peptide fragment of GIP1. So here we see this, as I was talking before, I was talking about D motifs that bind to CD domains. Oh my god. Okay, so I was talking about D motifs that bind to CD domains. So here we have a D motif. So we're going to have a leucine, which is going to represent the phi A. We have the asparagine, which is going to represent the X. And we have leucine 162, which is going to represent the phi B. So these 3 bolded residues are all going to be a part of the D motif of GIP1. So the D motif of GIP1 is going to bind to the CD domain of J&K1. So all the residues on GIP1 are kind of going to be enveloped by a hydrophobic region on J&K1, and these are the specific residues that form generally hydrogen bonds between the residues that they line up with. So again, proline would form hydrophobic bonds with valine, and so on and so forth. So I just listed them there. Hopefully this slide is helpful. If we don't like it, we can take it out. No big deal. So I guess this needs kind of more so of an introduction slide, kind of saying, now we're going to get into talking more so about why we want to inhibit J&K1 and how it exerts its effects on IRS1. Just want to check the time here for how I'm doing. I don't even know how.